Antimicrobial resistance is becoming a bigger and bigger concern within the medical community, but also throughout the population as well. It's been a priority in the research and medical field for quite a while now, but more recently there's also been a shift in attention towards making sure the public understand its importance, particularly when it comes to the case of differentiating between antimicrobial and antibiotic resistance. The UK Health Security Agency in particular are really pushing for better public literacy around antibiotic use, which is the focus of their recent digital campaign with antibiotic as the figurehead, which we'll get into more later. But to understand what happens when bacteria are resistant to antibiotics, we need to start with how antibiotics were designed to work in the first place before resistance became such a big part of the equation. Gab, this is an area you know pretty well, thanks to your research at the Quadram Institute at the University of East Anglia. Could you take us back to the beginning? How do antibiotics actually work?
When there is a bacterial infection and it becomes resistant to antibiotics that's used to treat it, it basically makes treatment times longer, it makes treatments more difficult, and it makes surgeries riskier. So a lot of people tend to mix up antimicrobials and antibiotics, but those are actually two completely different things.
Okay.
So antimicrobials are this umbrella term. It's basically any substance or compound or drug that can kill a microorganism in general. This would be things like antifungals, antivirals, and antibiotics. Things that can kill or inhibit the growth of fungi, viruses, bacteria, and a range of other microbes. Antibiotics are specifically for bacteria. So these are any chemical substance or drug that can kill or stop the growth of bacteria. All antibiotics are antimicrobials, but not all antimicrobials are antibiotics.
Sure. So you can kind of think of bacteria as a type of microbe, so that you know that which one kind of is the umbrella term, and then antibiotics are a subsidiary of that.
That's exactly it.
Cool.
Yeah. So from the beginning...
Let's go back.
Let's go back right to the beginning. Well, when I was in university, they taught us a lot about antimicrobial resistance and bacteria in general. And there was this term that I picked up called bacterial warfare. So microorganisms exist in this complex community with other microorganisms. And they have loads of different survival mechanisms that allow them to cooperate and establish relationships. But they also have mechanisms that allow them to compete. So there are things like space and nutrients that they need to fight for. Microorganisms in general can develop survival mechanisms to kill other microbes. One of these things are antimicrobials. And to defend themselves, bacteria can develop antimicrobial resistance. A really interesting example of this is the penicillium fungus. So Alexander Fleming discovered penicillin, and it was because of a fungus. I can talk more about that later, but it's basically been this intrinsic defense mechanism for ages. Actually, there was this interesting study in 2011. It was a Nature paper where they basically dug out a massive tube of ice, permafrost, from the Siberian region. And it was basically 30,000-year-old permafrost. And they found that there were genes encoding resistances to several major antibiotic classes in 30,000-year-old permafrost.
Oh, wow. So this is not like a recent development.
It's been there for ages. Well, this suggests thousands of years, but it may actually date back further than that. So I study phylogenetics, which is the evolutionary history of a group of organisms, but we can actually extend our studies past that and look at the evolution of certain genes. And we can look at these AMR genes and actually find out how far back they dated. A really interesting study in 2002 estimated that a specific AMR gene called a beta-lactamase gene are possibly billions of years old.
We can trace genes linked to resistance back a really long time, but when did we see the first antibiotics?
The very first antibiotic technically wasn't penicillin, it was salvarsan, which was used to treat syphilis.
Okay.
In 1928, that's when Alexander Fleming did his very classical experiment. You know the story?
Yeah, it's a classic science story.
For those who don't know, basically, Alexander Fleming basically was working with this Staphylococcus bacteria in his lab. And he decided to go on holiday because...
As you do.
Why not?
That's what he deserves.
Work-life balance.
Yeah.
That's what we want. He left for holiday, and then when he came back, he found that there was mold growing on his plates where the staphylococcus was growing. And he found that around this mold was a zone, basically a zone of inhibition where the microbes weren't growing. He then isolated the compound that was causing this inhibition, and that's where penicillin came from. After then, we kind of had this golden age of antibiotic discovery from the 1940s to around the 1960s. But we knew about antibiotic resistance since before then. So there was this interview by Dr. Bill Franklin, who was working with Fleming at Imperial College London, and he recalls that Fleming warned about the overuse and misuse of the drug already.
Oh, wow. So even back
when he
way
first
back
discovered,
when, it was always known that it was going to develop into maybe not the scale of the issue it is
right now,
but it was always kind of there in their minds.
Exactly. But it was actually in 1945 at his Nobel lecture where Fleming actually told the whole scientific community, he warned them about the misuse and overuse of antibiotics. So we've known about this for ages now. Unfortunately, because of the slowing down of novel antibiotic drug discovery, we're now in this discovery void. Nothing new is being produced, and this is a major issue for us. At the same time, there was this really big problem brewing, the antimicrobial resistance crisis that we have right now.
Your research focuses on E. coli specifically. Where did this bacteria come from?
For me, E. coli is such an interesting organism. It was first discovered by a German physician. He was a pediatric physician called Theodor Escherich in 1885. And initially, he called it Bacterium coli commune or Bacterium coli communis. And he isolated it from the feces of newborn babies.
Oh, wow.
Yeah. So he was a pediatric physician, and he was just working in hospitals, and that's where he was getting these samples from. And E. coli is commonly found within the guts of humans, but also a range of different animals. So you can find them in the guts of companion animals, in bovine creatures, ovine creatures, in a range of different things. And because of this, E. coli is typically used as a fecal contamination indicator, as well as a sentinel organism for AMR. So what I mean by this is that they're used to kind of scan for the presence of certain AMR genes.
Okay, so it's kind of used as an indicator.
Exactly. E. coli can fall into a couple different categories. You get commensal E. coli. So these are the ones that can live happily within our guts and not necessarily cause any intrinsic damage or acute harm. Then we have the more dangerous ones. These ones can be pathogenic. So we have different pathogenic types or pathotypes of E. coli. But also E. coli can be opportunistic pathogens, which means that if they get into the wrong place at the right time, they can cause some really serious infections. So E. coli can cause gastrointestinal disease. It's one of the organisms that we know can do that. But it's only a select few. So I talked about how some can be commensal, some can be pathogenic, and then cause immediate harm. When someone has a GI infection and it's from E. coli, we typically don't use antibiotics to treat it.
Okay.
We actually just use fluids and rest. And some studies suggest that actually using antibiotics on certain pathogenic lineages of E. coli can make problems worse. So an example that I like to use is Shiga toxin-producing E. coli. It produces this toxin that can be quite dangerous to us. Mm-hmm. It can cause symptoms like diarrhea, vomiting, but also bloody diarrhea.
Okay.
And hemolytic uremic syndrome as well. For things like that, clinicians typically don't prescribe antibiotics. However, when it's an opportunistic infection, for example, a bloodstream infection or a urinary tract infection, that's when we can treat it with antibiotics. For bloodstream infections, it depends on a couple factors. So, for example, if the E. coli are already resistant to certain antimicrobials. For urinary tract infections, it also depends on a couple different factors. So, for example, it depends on biological sex and pregnancy status, and also whether they are susceptible to certain antimicrobials. For UTIs, it's typically nitrofurantoin or trimethoprim, but then there are a range of other antimicrobials that we can use.
A common way to pick up an E. coli infection is by eating contaminated food. Food was, of course, the topic of the 2024 Christmas lectures with Chris Van Tulleken. And in the third lecture, Chris mentioned something that really kicked our development up a gear as a species, particularly for our brain development, was when we started to cook our food. Is E. coli one of those bacteria that cooking our food helps to kill?
Yes. Yes, it is. So one thing to note is that retail foods are not a sterile space. They can be contaminated by a range of different microorganisms. And these organisms can cause gastrointestinal disease, but they can also just be present on the food and not cause GI infections. There are organisms on there that can cause food spoilage, for example. So for cooking food, there's actually a UK government agency responsible for protecting public health in relation to food called the Food Standards Agency. And they have an E. coli cross-contamination guidance online. It's really good information. It's about preventing cross-contamination, how we can cook food correctly, and also about good personal hygiene. So, for example, you don't want to be handling raw meat with your hands, and you want to be changing your chopping board every so often, washing that thoroughly. And there are lots of rules and regulations set in place to monitor and survey contamination on retail foods. These are set into our law, not just for E. coli, but for a range of other really important relevant bacteria that cause gastrointestinal infections, and even other microorganisms outside of bacteria. So we also look out for viruses on food as well.
We've learnt already that there are two broad different types of E. coli, ones that are dangerous for us and ones that aren't. When these exist in the ecosystem of food or a supermarket, how can we differentiate between the good and the bad ones?
No, so you're right. Not all E. coli can cause acute gastrointestinal disease. And telling the difference between what can and can't cause disease can actually be a bit more of a difficult question. So one of the factors we need to take into account are the different types of E. coli. So we have those pathogenic types that I was mentioning earlier, but we can actually characterize them further into different pathogenic types of E. coli. Each of these have their own clinical presentation. Some strains of E. coli only require a low number of cells to cause gastrointestinal disease. This is the case for STEC, so the sugar toxin producing E. coli. Some papers say as low as 100 cells can cause acute disease. And it's also reflected in our laws and guidelines as well. When we're trying to define what can and can't cause disease, this is what I was mentioning, where actually it depends on clinical presentation, right? So historically, we would have to look at how this presents in a person, and then we would classify it that way. But now we can look at the genes inside E. coli. We've found ways to identify certain genes that can be associated with certain E. coli infections. The difficulty there is that some of these genes can be mobile. So, for example, they can be present in different types of E. coli. And there are also hybrid pathotypes of E. coli, so that they can have multiple of these different kinds of virulence genes that are typically associated with one type. We also have the added complexity of different people presenting with different symptoms. So someone might be infected with a certain pathogenic type of E. coli and have really bad gastrointestinal disease, whilst another might be asymptomatic. And the mechanisms of why that is still kind of being investigated.
Whilst we're on a much more zoomed out level thinking about these infections, let's talk about the public understanding of AMR and why that's become such a focus for a lot of government organisations. There are some pretty scary stats going around about this. One that stuck out to me was that 39 million people could potentially lose their lives to antimicrobial resistance by 2050, which is over 1.5 million every year on average, which is a huge number. And whilst I was researching this episode, the UK Health Security Agency announced a new digital campaign which is hoping to educate young people specifically on their antibiotic use. What is the consensus on when we should be using them?
So that's a really good question, and I'm so glad that the UK HSA has this ad campaign to specifically target people around our age range. This message is about antimicrobial stewardship. So it's about this coordinated and organized approach to monitor the appropriate use of antimicrobials to basically keep them working. When should we be using antibiotics? When we know that it's a bacterial infection and when we are prescribed them. So I'm not a clinician, But I do know that the recommended guidelines say that we need to finish our full course of antibiotics, and we need to make sure that we're being prescribed the right kinds at the right time.
Yeah.
So, for example, for things like the common cold, we shouldn't be taking antibiotics because that's being caused by a virus. That's not going to cure it. So this has been highlighted in a bunch of different government programs and reports. So for example, there's the Veterinary Antimicrobial Resistance and Sales Surveillance Report, which looks at antibiotic sales, use, and AMR surveillance results in animals.
Okay.
We also have the PathSafe project, which ended in March earlier this year, and that tracks foodborne disease and AMR in the environment and the agri-food system. It really highlights this One Health approach, which is this collaborative and cross-disciplinary approach for tackling health issues like AMR.
The UK is obviously taking a particular focus on this. Are we seeing these pushes and these campaigns in other countries as well?
Well, the World Health Organization has highlighted this as a major global issue, which requires a lot of cross-disciplinary collaboration to occur. A lot of countries do have this as a focus for surveillance, and also we need to start working together to address this issue on a global level.
At an individual level, we can help this problem by knowing when to use antibiotics and when they're not relevant to use. What is the actual mechanism of how this increased or overuse of antibiotics spurs on their resistance?
Any kind of level of antibiotic use can facilitate this issue of AMR production. So, regardless of whether it is misuse, overuse, this can lead to a selection pressure. So, E. coli, let's say there is this ancestral lineage, a very past version of E. coli. And E. coli are one of those organisms that expand and grow through binary fission. Basically, they make clones of themselves. When they make clones of themselves, they can actually mutate randomly. And these random mutations can be beneficial, but they can also be non-beneficial. And when there is something like a selection pressure, for example, antimicrobial resistance, this may lead to certain lineages of E. coli surviving. And this mechanism is natural. This is how microbes have evolved over time regardless. E. coli can gain resistances to antimicrobials through two ways. The first way would be mutating the genes that they already have. An example of this would be quinolone resistance. So quinolones are an antibiotic that inhibit enzymes involved in DNA replication. And there are certain resistant E. coli that have evolved mutations in those kinds of genes, in this quinolone resistance determining region. Another way E. coli can gain resistance to antibiotics is by acquiring AMR genes through processes like horizontal gene transfer. These are things like transposons, also known as jumping genes, bacteriophage, which are viruses specifically infecting bacteria, and plasmids, which are small circular DNA molecules that can harbor different genes, for example, AMR genes. And E. coli are just naturally very good at acquiring these mobile genetic elements. And they can acquire it from other E. coli, the environment, and then also other bacteria and other species.
So you mentioned when we were talking about the different types of E. coli that you see, say, in food in a shop, and that there are some circumstances where even just 100 of these bacteria could be really detrimental. How is it that we actually go about detecting and measuring bacteria like E. coli and actually counting? Is it a case of counting those individual bacteria, or are there more complex, if you like, techniques that have developed to kind of make that process a bit easier?
Currently, we use enumeration techniques. So basically, we count or estimate the amount of E. coli that are contaminating certain retail foods. There are these colony-forming units that we look at on plates. So basically, we count the number of little tiny colonies, E. coli colonies. And that's how we estimate it. Once we have that kind of estimated amount, there are safe thresholds that are set by law. Anything above a certain amount of contamination is unsafe for human consumption. What we can also do is use biochemical tests just to confirm that these organisms are what we think they are. And we can use biochemical tests to see which organisms they are, and then we can do some further tests to look specifically for shigotoxin, for that STEC that I was mentioning earlier. Right now, there's some really exciting work that we're doing using whole genome sequencing. This is where we can get all the genetic information from an organism, and then we can examine all that DNA using sequencing techniques. Whole genome sequencing, or WGS, isn't typically used for routine detection on food, but it has been used in E. coli outbreak studies. It's now becoming increasingly more accessible, and its history of the development of whole genome sequencing is actually quite interesting. In the early 1980s, the original Sanger method involved 4 sequencing reactions in separate tubes using radio-labeled primers. These were 4 dyes that emit different fluorescence. And then we can use that to kind of decipher the whole genome sequence.
Oh, so it's a very visual technique.
Exactly. Right now, we have next-generation sequencing techniques. And actually, we've progressed this further, and now we have third-generation sequencing techniques, where we can now have longer sequences than previous. And these methods are incredibly useful for investigating the whole genomes of bacteria. Do you want to know the history about the first bacteria we sequenced?
You already know the answer to that question. Of
Heck
course
yeah.
I do.
Heck yeah. So the first bacteria that we sequenced was Haemophilus influenzae, and this was termed a revolution for studying microorganisms. The first bacterial genome we sequenced was in 1995. 30 years on, we're now sequencing thousands and thousands of genomes all at once. To put this into context, it took 13 years to sequence the human genome. Right now, we can generate that information in 5 hours.
Wow. Okay. So is this the kind of area that's seen the most exponential development?
I think this is one of the areas which has exponentially grown.
Yeah.
We're now developing new techniques, faster techniques, more efficient techniques. And it's very exciting because now we have all this genomic data that we're analyzing. That's basically my role as a PhD student. I do a lot of bioinformatics, so it's a lot of data analysis, a lot of genomic analysis as well.
What does using whole genome sequencing look like in practice?
So, for example, with my bacteria like E. coli, we can analyze the genomes of a large amount of E. coli from a range of different environments and hosts. From this, we can look at different populations and start to look at their evolutionary history, specific genes that are associated with specific places, and potentially how these E. coli have adapted to these places. E. coli is extremely genetically variable, and whole genome sequencing is an incredible tool for investigating E. coli populations. We've actually found that E. coli have this open pangenome. So this means that whenever we sequence a new E. coli and we add that to the current data set that we have, new genes are likely to pop up.
Whenever I hear people talking about vast amounts of data now, I just have this voice in my head going, is this something AI could help with? And I'm sure other people have that as well. So where does AI come into this field, if it does at all?
That's a really good question. So large language models are being used as a potential tool to analyze these genomes. It really depends on the question that you're trying to answer.
Sure.
I won't get into machine learning and AI right now, but there is actually an AI Bio UK community. So this is a BBSRC-funded network to support and enhance engagement between bioscience and AI communities in the UK. So these are a network of scientists who are using AI to look at how we can apply them to certain questions and answer really interesting things about the genomic information that we currently have.
As I'm sure people listening can tell, you're not new to communicating your research in a way that actually makes sense to people and is interesting and relevant. You also recently won a Creator Award. Congratulations.
Thank you.
And have previously spoken at the RI here in the theatre, last time you were here, for our event that was celebrating LGBTQ+ in STEM Day. How has science communication become a focus for you? And is it always something that you've been interested in doing alongside your research?
Science communication to me has always been a joy. I love yapping to people, telling people things that I've learned. I get to talk about cool sciencey stuff at work and then bring that home and then talk about cool sciencey stuff with my family. It's like teaching, but in like a really accessible format.
Mm-hmm.
I've always been like a really creative person, so I've always been making videos and pictures and stuff like that.
Yeah.
And this was just one way of blending my worlds together. It was something that I just really enjoyed. It came really naturally to me.
I think people also often try to separate science and the more, I hate using the word creative in this way, but the more kind of creative fields. But I think there's such a fluid, I don't think it's something you can separate. I think both science is in everything, and I am always the person to say, whatever your job is, you're doing and using science, because it's more of a principle and a way of going about things than actually sitting down and learning about cells. Sometimes it is that, but a lot of the time it's not. So I think it's always a process of using creativity.
I agree with you 100%. Everything has a scientific basis behind it. But I also agree that everything has a potential to be creative as well. You're right in the sense that science and creativity have always gone hand in hand. And this is a hill that I'm going to die on.
Yeah, me too. I'll be there with you.
To be at the forefront of science, to innovate, to create new things, you intrinsically have to be creative. You have to push the boundaries, and you have to build on the backs of giants. And this is what we're doing right now with the current research. So we are building on what we know previously, what these amazing scientists like Michael Faraday have done, and we're now innovating. We're being creative. Every scientist, I believe, is a creative at heart. We also have things like social media, where we have some amazing scientists, mathematicians, physicists, showcasing their uniqueness and talents online. And I think it's just a fantastic way to make things accessible, especially in this digital age.
So I mentioned that the last time you were here was speaking as part of our LGBTQ+ in STEM Day, which we did in collaboration with Pride in STEM. How important to you has representation been in science?
Extremely important. So growing up previously, when we've asked young people and children what their idea of what a scientist was, there would always be this kind of stereotype, this preconceived idea of what that is. Typically, you'd think of the scientists from Back to the Future, right?
Or a bit of an Einstein type
Yeah.
character.
Like the mad scientist. It would typically be a white, non-disabled, older male.
Yeah.
And for a lot of people, that isn't us.
Yeah.
And like many others, I've never had a lot of role models that I could see myself in, right? And when you lack that visibility, your scope of what you think you can do narrows. This is why I think things like Pride in STEM and other initiatives like What is a Scientist, 500 Queer Scientists, these things are important for showing what LGBTQ+ people can do, right? It's showing that this is a community that's accepting regardless. We are focused on the work, on the science, but we're also proud of who we are as people. I think there's this beautiful way of kind of looking at it where you are a culmination of every single aspect of you. So if you are a scientist, if you are gay, if you are a person of color, and you are all of those things, you are unique and you add value to the community, no matter what community that is.
I think science is all areas. This applies to all areas, but science, as that's what we're talking about, is so reliant on different perspectives, and all of that changes depending on where you've grown up and what community you've grown up in. And science isn't going to go anywhere if it's just in an echo chamber of people who've had the same experiences. So I think bringing those people with different experiences benefits science. It's purely thinking from a scientific progression point of view. We need it. It's important.
You're absolutely right. We need this, right? We need other people's perspectives, other outlooks. This is how innovation and creativity happens. We share knowledge. And by restricting that to a certain group of people, we're not going to have the same level of innovation that we're seeing right now.
Gab, I think we could yap all day.
Yes.
And we yapped a lot before we started recording. So we've saved you from a lot of that yap. But as our regular listeners will know, I like to finish with a bit of a rogue on-the-spot question, which is in the context of what we've been talking about, but gives us a bit of a deeper insight into your psyche.
Okay.
And you have not seen this question.
No, I have not.
So it was redacted out of your script. But my question for you is, I'd also like to prerequisite this question. You can't say E. coli, because that That's too obvious an answer. If you could be any bacteria, what would you be and why?
Fantastic question.
Thank you.
Am I allowed to have multiple answers?
If you can justify them, then yes.
There's a part of me that wants to be a beneficial bacteria.
Okay.
One that's part of your gut microbiome.
Sure.
So that's your collection of microorganisms, viruses, fungi, bacteria in your gut. I think one of those microbes, one of those microbes is bifidobacterium.
Mm-hmm.
And that's typically quite a healthy commensal. So that's found within the guts of a range of different healthy humans. But I think I want to be edgier than that.
Okay.
Yeah. Do I want to be a bacteria that causes major disease?
I think yes.
Oh.
Maybe. Well, maybe.
Well, recently I've been reading John Green's new book, Everything is Tuberculosis, and he highlights multi-drug resistant TB as a major global issue.
Okay.
So maybe MDR TB would be an interesting bacteria to be.
I mean, you want to leave your mark on the world, right? Why not go out with a bang and just wreak absolute havoc?
Well, with TB, actually, John Green says that that's why we have cowboy hats.
What?
Yeah.
Wait, what?
Yeah, that's how we have cowboy hats. Do you want to know why?
Yeah.
Yeah. So I'm paraphrasing. He's said this a couple times, so he's probably the best person. But basically, the person who designed the cowboy hat had TB. And during those times, they're basically told by the doctor to leave highly densely populated places and go to more rural places. This person was one of the lucky few that survived their TB infection. But he was now living in the rural area of US, and he saw that people were wearing terrible hats. These were like coonskin hats.
Oh.
I know. And they did not protect them, and it didn't protect them from the sun.
No benefits then
No
to
benefits.
those hats.
And then he was like, hey man, let me design the cowboy hat.
No way.
Yeah. So TB helped make the cowboy hat.
That is a better answer to this question than I ever could have imagined.
Thank John Green.
Thank you, John Green, for facilitating that answer. I think that's a wonderful note to end on. So, Gab, thank you so much for joining me.
Thank you so much for having me, Lia. It was a pleasure to chat with you.
If you want to find out more about the UK HSA's digital campaign about antibiotic resistance or enjoy more science from Gab, I'd recommend doing both. We've got links to everything you need in the description of this episode. And as always, if you want to enjoy more science from the RI, either online or here in the theatre, head to rigb.org to see what we've got coming up.